Hello students, this question has many sub-questions. So let us first answer the first question. The three structures of protein are primary structure. So the primary structure of protein consists of amino acid sequences. It is the primary structure of protein which gives the protein its features or characteristics. Then we have the secondary structure. The secondary structure is the folding of the primary structure. So when the primary structure folds, it gives us the secondary structure or the polypeptide, and there are two types, alpha helix and beta pleated sheets. The third type is the tertiary structure. The tertiary structure is the three-dimensional structure of a polypeptide. This is what gives us the protein structure. So after the amino acid sequence has folded into the secondary structure, then the secondary structure folds further to give us the tertiary structure. Secondly, the shape of a protein determines its function because its shape is determined by its amino acid sequence. Therefore, if the shape is determined by the amino acid sequence, it would also determine its function. And why is this important? It is important because proteins are specific, meaning that it interacts only with specific molecules. That is why its shape determines its function due to the amino acid sequence, which makes them to be specific in there. Now coming to the third question, the proton sucrose co-transporter, how it functions is that it uses ATP to generate proton out of the cell against the concentration gradient. Then the proton gradient that is generated is being used by the proton sucrose transporter to transport proton and sucrose to the cell. So let us recap. The co-transporter uses ATP to generate proton out of the cell against the concentration gradient. Then due to the difference in the concentration gradient, protons are transported back into the cell along with glucose. Coming to the fourth question, sucrose, being a large molecule, cannot pass through the membrane directly. How it passes through the membrane? It is through the process of endocytosis. During this process, what happens is that a part plasma membrane engulfs the large molecule and brings it inside the cell. Now coming to the next question, wherein we have to define the induced fit model of enzyme through the help of a diagram. So what happens in this induced fit model is that the enzyme, before binding to the substrate, has a different shape, but once it binds to the substrate, the shape of the enzyme changes. As you can see here, the shape changes, and this shape will fit perfectly the substrate. And as the substrate changes into the product, then the product will be released from the enzyme, and the enzyme once again will have its original shape. So this is induced fit model. Coming to the next question, wherein we have to explain with the help of a diagram the competitive inhibition. So what happens in competitive inhibition is that a competitive inhibitor has similar shape as that of the substrate. So as a result, the inhibitor will bind to the enzyme, forming less enzyme substrate complex, thereby decreasing the product formation and decreasing the overall rate of enzymatic reaction. So as you can see in this picture, we have the enzyme and the inhibitor. The inhibitor has a similar shape as the substrate, so it will bind, thereby preventing the substrate to bind, forming less products and in turn decreasing the rate of the reaction. Hope you found the video useful. Thank you.